Hey everyone, Dr. Steve here. You know it as well as I do, but most students need a B or better in A&P. But did you know that students are doing that all over the US using my Anatomy and Physiology Digital Suite as a low-cost alternative to expensive textbooks? Digital Suite delivers the key concepts of A&P with short, assignable tutor videos and a bulleted narrative study guide that not only explain the concepts of A&P, they show them happening. Combined with state-of-the-art dissection and physiology lab simulations and a vast question bank for quizzes, homework assignments, and exams, Digital Suite meets students where they are and gives them the tools to master the content. It's the perfect supplement to an OER textbook or just on its own. If you're an A&P instructor, contact your McGraw-Hill representative today and ask about Anatomy and Physiology Digital Suite. If you're a student, tell your instructor about it. There's a link to Digital Suite in the show notes of this episode. Get your B or better in A&P with A&P Digital Suite.
At 1-800-Flowers.com, we know that connections are at the heart of being human. Whether celebrating life's joys or comforting during tough times, 1-800-Flowers helps you express what words can't. For nearly 50 years, millions have trusted 1-800-Flowers to deliver thoughtful gifts that help create lasting bonds. Because it's more than just a gift. It's your way of showing you care. Visit 1-800-Flowers.com/WWO and connect today. That's 1-800-Flowers.com/WWO.
This episode is brought to you by Progressive Insurance. Fiscally responsible, financial geniuses, monetary magicians. These are things people say about drivers who switch their car insurance to Progressive and save hundreds. Because Progressive offers discounts for paying in full, owning a home, and more. Plus, you can count on their great customer service to help you when you need it, so your dollar goes a long way. Visit Progressive.com to see if you could save on car insurance. Progressive Casualty Insurance Company and Affiliates. Potential savings will vary. Not available in all states or situations.
Suffering from dry, tired, irritated eyes? Don't let dry eyes win. Use Systane Pro. It hydrates, restores, and protects dry eyes for up to 12 hours. Systane Pro. Triple action dry eye relief.
Hey everyone, welcome back to my podcast, Anatomy and Physiology Bit by Bit. I'm your host, Dr. Steve Sullivan, coming to you from Bucks County, Pennsylvania, where I've been teaching A&P since 2002. Today's episode is about genetics and heredity. I'm going to stick to the level of genetics that you typically get with a 100-level A&P course, so it should be applicable to most of you listening, and accessible to pretty much anyone. Before we get started, I want to take a minute to let you know about a really fun podcast that I think you're going to like, especially if you like animals and you have kids. It's called Smarty Pants Wonders, and it's hosted by Dr. Tabitha Delangelo. Every episode is about a different animal with an expert guest sharing pretty much everything they know about that animal. There have been episodes about turtles, foxes, lanternflies, butterflies, and more. It's really fun for kids. And honestly, I like it. I have learned more than I thought I could about animals that I see running around my backyard every day, like squirrels. I put a link to the show in the show notes of this episode so you can check it out for yourself. I hope you like it as much as I do. Having said that, let's get going with today's topic. Heredity is, at its core, a story about continuity. Every person alive carries a biological archive of information that stretches back through time, encoded in microscopic structures inside their cells. When parents conceive a child, they do more than contribute cells. They hand over instructions, recipes for building a new human being. Those instructions are what we call heredity, the transmission of genetic characteristics from one generation to the next. To understand how traits run in families, how diseases can appear, disappear, and reappear across generations, and how something as complex as cancer can have its roots in tiny changes in DNA, we need to start with the basic players: chromosomes, genes, and alleles. Chromosomes are the packages of heredity. Inside the nucleus of almost every cell in your body are structures called chromosomes. These are long strands of DNA coiled and folded around proteins packed tightly enough to fit into a microscopic space, yet carrying staggering amounts of information. If we take the 46 chromosomes in a human cell, stain them, and arrange them in order by size and other features, we get a picture known as a karyotype. It looks like 23 pairs of X-shaped or rod-like units lined up in neat rows. Each pair in that karyotype is a pair of homologous chromosomes. 2 chromosomes that are similar in size, shape, and gene content. 1 chromosome of each pair came from your biological mother from her egg cell, and the other came from your biological father in his sperm. With one important exception, the 2 homologous chromosomes closely resemble each other and carry the same genes in the same locations, although the exact versions of those genes can differ. That exception is the pair known as the sex chromosomes. These are labeled X and Y. The other 22 pairs of chromosomes are called autosomes, and both members of each autosomal pair look alike. In biological females, the sex chromosomes are typically a homologous pair of X chromosomes, XX. In biological males, the pair consists of 1 X chromosome and 1 smaller Y chromosome, XY. The combination of sex chromosomes largely determines whether the developing embryo follows a typical biological male or biological female pathway. Although, as we'll see later, this is not always strictly binary. A cell that contains 23 pairs of chromosomes, 46 total, is described as diploid, symbolized as 2N. The reason for this is because ploidy is the number of chromosomes that a species has. Humans have 23 different chromosomes, and we have 1 from each of our biological parents, so 46 total. So if our ploidy is 23, then we will use the letter N to represent 23. So diploid, or 23 pairs, would be 2N. Almost all of your body's cells are somatic cells, which are diploid. By contrast, the cells that give rise to sperm and eggs, called germ cells or sex cells, undergo a special kind of cell division called meiosis, which we've talked about in a previous episode. In fact, episode 69. In meiosis, the homologous chromosomes in each pair are separated so that the resulting sperm or egg only contains 1 chromosome from each pair, for a total of 23 unpaired chromosomes. Since that cell only has 1 of each, it is haploid, and we symbolize that with just the letter N. At fertilization, a haploid sperm cell from the biological father unites with a haploid egg cell from the biological mother. Their chromosomes combine, restoring the diploid number in the resulting fertilized egg or zygote. From that single cell, countless rounds of cell division produce the trillions of somatic cells that make up the body, each carrying the same diploid set of chromosomes derived from that first union. Once again, you can review episodes 72 and 73 to hear about embryology and human development, and that will review the details of what I just summarized. So heredity depends on 2 things happening in sequence. Meiosis, which separates the pairs of chromosomes into haploid gametes, and fertilization, which brings together 1 set from each biological parent, recreating the diploid condition. Within those chromosomes are the actual units of heredity, the genes. So let's talk about genes and alleles, which is the language of inheritance. A gene is a stretch of DNA that contains instructions for building a specific product, often a protein that performs some function in the cell or body. These are called traits, with examples being eye color, hair color, or whether or not you can accurately make a particular enzyme, like lactase for breaking down lactose, which is known as milk sugar. Thanks to the Human Genome Project and its head, Francis Collins, and later research, the positions of many genes on the chromosomes have been mapped. The specific location of a gene on a chromosome is called its locus, with the plural being loci. Homologous chromosomes carry the same genes at the same loci. However, the exact versions of those genes can differ. These different forms are called alleles. Alleles of a single gene may produce slightly different versions of the protein associated with a trait. It may even result in a non-functional variant of the protein. That in turn can lead to visible differences like eye color, or subtle biochemical differences like how we process cholesterol, for example. Often one allele is described as dominant and the other as recessive. If at least one chromosome in a pair carries the dominant allele, the trait associated with that allele is usually expressed, meaning it shows up in the organism's observable characteristics, which we call the phenotype. The recessive allele's effect is masked in this situation. Typically, if a dominant allele codes for a functional protein, a recessive allele might code for a protein that is altered or non-functional. Because the dominant allele produces enough functional protein to carry out the job, the recessive allele's effect is hidden unless both copies are recessive. The combination of alleles a gene has for a given trait is their genotype. For example, if we use the capital letter A for a dominant allele and the lowercase letter a for a recessive allele, possible genotypes are capital A, which we call homozygous dominant. Since the prefix homo means same, this means the gene has two copies of the dominant allele for that trait. Then we have lowercase a. This is homozygous recessive, meaning two copies of the recessive allele. Then finally, there's capital A and lowercase a, heterozygous. Hetero means different, so we have one dominant allele and one recessive allele for that trait. The genotype describes what's on the genes. The phenotype describes how those genes actually show up in the organism. Eye color, presence or absence of a disease, blood type, enzyme activity, and so on. We say an allele is expressed if it has an observable effect in the phenotype, whether that is something obvious, like brown versus blue eyes, or something that requires laboratory testing, like the presence of a particular enzyme in the blood. A good example of something genetically passed on is sickle cell disease. This is something that is located on a single gene, but with very big consequences. Now, although many traits are influenced by multiple genes, some important conditions like sickle cell disease are determined chiefly by a single gene with dominant and recessive alleles. Millions of people around the world live with disorders that can be traced largely to a single mutated allele. Examples are cystic fibrosis, Huntington disease, certain muscular dystrophies, and then sickle cell disease, like I've mentioned. These are among the most well-known. I want to talk about sickle cell disease to really bring this point home. It's a good example because it shows how a small change in a gene can have drastic effects on the body, and even on the evolution of entire populations. Recall from my previous episodes on blood, where I talk about red blood cells, hemoglobin, etc., and this would be episode 39, that hemoglobin is the protein in red blood cells that binds oxygen and transports it from the lungs to the tissues. Remember, red blood cells are known as erythrocytes. Normal adult hemoglobin is called HbA. In sickle cell disease, a recessive allele produces an abnormal hemoglobin variant called HbS. HbS differs from HbA in just one amino acid in the beta chain of the protein. So, where HbA has glutamic acid, HbS has valine at the 6th position. That tiny substitution dramatically alters the behavior of hemoglobin under low oxygen conditions. Now, at normal or high oxygen levels, HbS behaves somewhat like normal hemoglobin. But when oxygen concentrations are low, as in smaller veins or tissues where oxygen is being unloaded, HbS becomes deoxygenated and tends to polymerize, meaning the molecules stick together and form long, rigid rods. These rods distort the red blood cells into elongated, curved forms with pointed ends, kind of like the farmer's sickle. The affected cells are called sickled erythrocytes. These sickled cells are stiff and sticky. They tend to clump together and block small blood vessels, cutting off blood flow to the tissues that they supply. This leads to episodes of intense pain, especially in bones and joints, as well as damage to organs like the kidneys, heart, and brain. Repeated vessel blockages can cause strokes, heart failure, paralysis, and other serious complications. And because the sickled cells are fragile, they rupture easily. We remember that from those episodes on blood called hemolysis, which can lead to anemia, which is a shortage of functional red blood cells. Anemia, in general, means less oxygen is being delivered to tissues, and therefore hypoxemia, or low blood oxygen, is the result. Ironically, hypoxemia further encourages hemoglobin to deoxygenate and sickle, creating a vicious positive feedback loop. Low oxygen causes more sickling, which worsens circulation and oxygen delivery, which causes more sickling, and so on and so on. Over time, chronic hypoxemia leads to fatigue, weakness, delayed growth and mental development in children, and progressive damage to the heart and other organs. In response to chronic anemia, the body tries to ramp up red blood cell production. Bone marrow and other blood-forming tissues become overactive, causing expansion and distortion of bones, particularly in the skull and other flat bones. The spleen, which normally filters out old red blood cells, becomes packed with dead and damaged erythrocytes, enlarges, and may become fibrotic. Without treatment, children with sickle cell disease often die early in childhood. Modern medical care has increased life expectancy, but even with treatment, the disease can shorten someone's life by decades. So why then would such a harmful gene even persist in our population? Well, that is the genetic trade-off of sickle cell disease. You see, sickle cell disease is especially common in people whose ancestry lies in regions where malaria has been historically widespread. Places like sub-Saharan Africa, parts of the Mediterranean, the Middle East, and India. Malaria is caused by a parasite called Plasmodium that invades red blood cells and feeds on their hemoglobin. In individuals who are heterozygous for the sickle cell allele, meaning they carry one normal hemoglobin allele and one sickle cell allele, the red blood cells contain both HbA and HbS hemoglobin. These heterozygous individuals don't usually develop full-blown sickle cell disease. Their red blood cells may sickle mildly under extreme conditions, but most of the time they function well enough that the person does not experience the severe anemia and other crises of someone who is homozygous for the HbS allele. However, their slightly altered red blood cell environment is less hospitable to the malaria parasite. The infected sickling red blood cells are more likely to be destroyed, reducing parasite survival and not allowing it to multiply. So in malaria endemic regions, this creates a powerful evolutionary advantage for heterozygous individuals. They have greater resistance to malaria and are more likely to survive and pass their genes on to their children. The lives saved by the heterozygous carriers of the sickle cell gene more than offset the deaths of the individuals that are homozygous for sickle cell disease. As a result, the sickle cell allele remains relatively common in these populations. Now, the downside is we have to look past the personal tragedy of the people who actually get sick from sickle cell disease, which is hard to do. Nobody wants to do that. And honestly, from an evolutionary perspective, that's what we're doing. Because ultimately, evolution doesn't care about the individual person. Evolution is not a perfect system. Evolution is cruel. And most species that have ever existed on the planet have gone extinct. But that is the case here, where because of the heterozygous nature of the dominance and recessive combinations, we end up with far more people who have a resistance to malaria than we have people who have sickle cell disease. And the resistance to malaria in those regions before there were anti-malarial medications was a far better survival advantage. And that's why we still have sickle cell disease. Now, in regions where malaria is rare, like North America and Europe, this evolutionary advantage disappears and the frequency of sickle cell alleles tends to be lower. This illustrates how the environment interacts with genes to shape the genetic composition of populations.
'Tis the season of gifting and halls to deck, and the Who's and Who Newville were in love with new tech. Where can we find Sonos and Samsung and Nintendo, they shouted. Would they find it in one place? This they questioned and doubted. When suddenly a Who yelled, Walmart's the place to start! And each Who added headphones, TVs, and games to their carts. With Walmart, their shopping was done in a flurry. They cried out, Who knew? And ordered their gifts in a hurry. Shop the latest tech gifts in the Walmart app.
Lowe's knows that saving is always top of mind, especially this season. That's why we've picked some great deals for early Black Friday. Get free select DeWalt, Cobalt, or Craftsman tools when you buy a select battery or combo kit. More tools? Why not? Plus, we've got select pre-lit artificial Christmas trees starting at $59.98 because it's never too early to think Christmas. Get Black Friday prices without the crowds. Lowe's, we help, you save. While supplies last, selection varies by location.
At Capella University, learning online doesn't mean learning alone. You'll get support from people who care about your success, like your enrollment specialist who gets to know you and the goals you'd like to achieve. You'll also get a designated academic coach who's with you throughout your entire program. Plus, career coaches are available to help you navigate your professional goals. A different future is closer than you think with Capella University. Learn more at capella.edu.
I'm sure you've heard the phrase skipping a generation when it comes to traits. To see how sickle cell disease can skip generations, geneticists often use a diagram called a Punnett square. Since drawing the square itself is futile in an audio podcast, we can walk through the logic in words. Let's use the capital letter H for the dominant allele that produces normal hemoglobin, and the lowercase h for the recessive sickle cell allele. The possible genotypes, therefore, are capital H, which is normal hemoglobin, no sickle cell disease, and cannot pass on the sickle cell allele to offspring. Capital H, lowercase h, this is a carrier of the sickle cell trait. They're heterozygous, and they usually have no disease, but can pass on the allele to their offspring. And finally, lowercase h, this is an individual who is homozygous recessive and has sickle cell disease. Imagine that both parents are heterozygous, capital H, lower h. Each parent produces gametes, sperm or eggs, that carry one allele each. In the biological mother, about half of her eggs carry capital H and half carry lowercase h. In the biological father, about half of his sperm carry capital H and half carry lowercase h. When we consider all possible pairings of sperm and egg, we get this. A capital H sperm fertilizes a capital H egg, and we get a capital H child. No sickle cell disease, doesn't even carry it. If a capital H sperm fertilizes a lowercase h egg, we get capital H lowercase h in the child. That child will likely not have the disease, but be a carrier of the trait and could potentially pass it on to their offspring. They're also immune to malaria. If a lowercase sperm fertilizes a capital H egg, we also get capital H lowercase h in the child. Same thing. Heterozygous, they carry the gene, they can pass it on to their kids, but they're likely not sick. And they're immune to malaria. Finally, a lowercase h sperm fertilizes a lowercase h egg. We have a homozygous recessive lowercase h child, and unfortunately, that individual will have sickle cell disease. Statistically, each child of this couple has a 25% chance of being capital H, normal hemoglobin, no sickle cell allele at all, but also no immunity to malaria. They have a 50% chance of being uppercase H, lowercase h. This carries the trait but not the disease and has the malaria immunity. And finally, a 25% chance of being lower H, and therefore having sickle cell disease. That 50% of people that could have an immunity to malaria is so huge in a region where malaria was killing an enormous number of people. Now notice that both of these biological parents may appear perfectly healthy, but each can carry the recessive allele. If their genotypes are uppercase H, lowercase h, they can have a child with sickle cell disease even though neither parent shows symptoms. In this way, a recessive disease can appear to skip a generation. It is present in the genotype of the parents, but hidden in the phenotype, only revealing itself in some of their children. Because of the profound impact of such inherited disorders, genetic counseling has become an important field. Genetic counselors help individuals and couples understand their risks of passing on inherited conditions. They may arrange for genetic testing to identify carriers, explain probabilities of inheritance, and help people cope with the medical and emotional implications of genetic diseases. Okay, it's not always that simple with a simple dominant and recessive allele. We have multiple alleles, we have codominance, we have incomplete dominance. So far we've only talked about genes with just two possible alleles, one dominant, one recessive. Real human genetics is much more complex. Some genes have multiple alleles within the population. This doesn't mean a single person has more than two alleles for a gene. Each individual still carries at most two versions, one on each homologous chromosome. But across the whole population, there can be many variants. A classic example is the ABO blood group. Again, I covered this in episode 39, and you might want to go review that. Remember from episode 39 that the ABO blood group is based on antigens that appear on erythrocytes. The antigens are the A antigen and the B antigen. The gene that determines ABO blood type has three primary alleles. There's capital letter I with superscript A, which codes for the A antigen on erythrocytes and is dominant. We have capital letter I and superscript B, which codes for the B antigen and is also dominant. And lowercase I represents the allele that produces neither the A nor the B antigen, and that one is recessive. Depending on which two alleles a person inherits, their genotype and blood type, which would be the phenotype in this case, will follow the following scenarios. Scenario 1. Both parents deliver the A antigen allele, or one parent does and the other delivers the allele for no antigen. Since type A is dominant, in this case the child will be type A blood type and produce the A antigen on their erythrocytes. So this would look like one of the following. Capital I superscript A, capital I superscript A. That would be one genotype. Two of the A antigen alleles. Or it could be capital I superscript A and lowercase I. That's the heterozygous version. And both of those scenarios, the child will have type A blood. Their erythrocytes will express the A antigen. Now also in scenario 2, we have both parents could deliver the B antigen, very similar. If they both deliver the B antigen allele to their offspring, or one parent gives the B antigen allele and the other delivers the allele for no antigen, then since type B is also dominant, in this case, the child will be type B and produce the B antigen on their erythrocytes. So uppercase I, superscript B, uppercase I, superscript B, that's the homozygous type B dominant version, or uppercase I, superscript B, and lowercase I, this is the heterozygous type B blood version. In either of these cases, the offspring will be type B and express the B antigen on their erythrocytes. Now here's where it gets tricky. If one parent delivers the A antigen allele and the other delivers the B antigen allele, the child's erythrocytes will have both the A and B antigen on them. And that will result in the child being type AB blood. In this case, both alleles are dominant, so both are expressed. This is codominance. Capital I superscript A, capital I superscript B. That results in type AB blood, and their red blood cells will have both A and B antigens on them. The final scenario is that both parents deliver the allele for no antigen, which is recessive. The child's erythrocytes will express neither antigen and be type O. Lowercase I. That's type O blood, no antigens. So that's codominance. Another pattern of inheritance is incomplete dominance, where the heterozygous phenotype is intermediate between the two homozygous phenotypes. One example in humans is familial hypercholesterolemia. This is a disorder involving LDL cholesterol, or low-density lipoprotein, and its receptor function. People who inherit a defective allele from both parents, meaning it's homozygous, can have cholesterol levels several times higher than normal and may suffer heart attacks as early as childhood. People who inherit just one defective allele and are heterozygous have elevated cholesterol. Instead of being several times higher, it's like 2 to 3 times normal. And they're at high risk for early heart disease, but typically later in life than the homozygotes. The heterozygous phenotype lies between the extremes of normal and severely affected, illustrating incomplete dominance. So we can also talk about polygenic inheritance and pleiotropy. Many traits don't follow simple one-gene patterns at all. Polygenic inheritance refers to traits controlled by genes at multiple loci, sometimes on different chromosomes. Each gene may contribute a small additive effect, and the combined contributions produce a wide range of phenotypes. Human skin color, hair color, and eye color are classic polygenic traits. Instead of being determined by a single brown eye gene or blue eye gene, these traits emerge from the interaction of dozens or even hundreds of genes. That's why there is such continuous variation in these traits, and why it can be difficult to predict a child's exact appearance based solely on the parents' traits. Many common diseases are also influenced by polygenic inheritance. Conditions such as some forms of alcoholism, certain mental illnesses, various cancers, and heart disease appear to reflect the combined action of multiple genes interacting with environmental factors. Each variant may contribute a small increase in risk, and the overall risk emerges from the sum of genetic and environmental influences. The flip side of many genes one trait is pleiotropy, where a single gene influences multiple traits or organ systems. A striking example is a rare genetic disorder called alkaptonuria. It is caused by a mutation on chromosome 3. This mutation interferes with the breakdown of the amino acid tyrosine, causing an accumulation of an intermediate compound called homogentisic acid in body fluids and connective tissues. As homogentisic acid oxidizes, it binds to collagen and causes tissues to darken to a gray or bluish-black color. Over time, this leads to multiple phenotypic effects: darkening of the skin and ear cartilage, discoloration of the sclera and teeth, dark urine that turns almost black when it's standing around, degeneration of joint cartilages with resulting arthritis in hips, knees, and spine, and damage to heart valves, the prostate, and other internal organs. From one mutated gene arise numerous clinical manifestations. This is pleiotropy. Sickle cell disease is another classic pleiotropic condition. That single amino acid substitution in hemoglobin leads not only to anemia, but to bone deformities, organ damage, susceptibility to infections, enlargement of the spleen, growth and development delays, and tons of stuff. Another important aspect of genetics is sex-linked inheritance. Not all genes are on the autosomes. Some are located on the sex chromosomes, particularly the X chromosome, which carries around 900 genes, many unrelated to sex determination. The Y chromosome is much smaller and carries relatively few genes, maybe around 55. And they mostly relate to testis development and male fertility. Traits controlled by genes on the X and Y chromosomes are called sex-linked traits. Most known sex-linked traits are X-linked simply because the X chromosome has so many more genes. Consider red-green color blindness or certain forms of hemophilia. The alleles responsible for these conditions are recessive and located on the X chromosome. A biological female has two X chromosomes, so if she inherits a recessive color blindness allele on one X chromosome, she may still inherit a normal dominant allele on the other. The dominant allele can mask the recessive, so she has normal color vision but is a carrier. A biological male, however, has only one X chromosome and one Y. If he inherits an X chromosome carrying the recessive allele from his mother, he will be colorblind because there is no second X carrying a normal allele to compensate for it. His Y chromosome doesn't carry a corresponding gene for color vision. This is why X-linked recessive traits are much more common in biological males than in biological females. Notice an interesting twist. A colorblind son can only inherit his X chromosome and therefore the colorblindness allele from his mother. His father contributes a Y chromosome, not an X. A daughter can be colorblind only if she inherits the recessive allele from both parents, from her mother on one X and from her father, who must himself be colorblind, since he has only one X. The combination is relatively uncommon, which is why colorblindness is rare among biological females. Although biological males do not receive an X chromosome from their fathers, there is still some genetic exchange between the X and Y chromosomes in the biological male during meiosis. In regions near their ends called pseudo-autosomal regions, the X and Y can swap segments of DNA. Occasionally, genes can move from the X to the Y or vice versa. For instance, the SRY gene, the key male-determining gene that initiates testis development, is normally on the Y chromosome. In rare cases, it can transfer to the X. This can result in individuals whose chromosomal sex, XX or XY, does not strictly match their gonadal or phenotypic sex, underscoring that sex determination is more complex than just X versus Y. It's easy to assume that dominant alleles are more common in the population than recessive ones. After all, doesn't dominance imply strength or prevalence? Well, in genetics, it doesn't. Dominance and recessiveness describe how alleles interact with individuals, not how frequent they are in the gene pool. For example, in North America, type O blood is the most common. Yet type O results from being homozygous for the recessive allele. By contrast, type AB blood, which requires two dominant alleles, is the rarest of the ABO blood types. Another example is polydactyly, the presence of extra fingers or toes. The allele for polydactyly is dominant, but the trait is relatively rare, occurring in roughly 1 in 700 to 1,000 births. A dominant allele can remain rare if it does not spread widely in the population, it reduces reproductive success, or simply by chance. So the frequency of an allele in a population depends on evolutionary forces, selection, mutation, migration, genetic drift, not on whether the allele is dominant or recessive. Even if a person has a particular genotype, that doesn't guarantee that the corresponding trait will appear exactly as expected. For a gene to have an observable effect, it must be expressed. Its instructions must be used to make RNA or protein in a cell. The extent to which a genotype leads to a phenotype is influenced by many factors. Sometimes a dominant allele is present but not fully expressed in every carrier. In such cases, we say the allele has incomplete penetrance. The genotype does not penetrate to the phenotype in everyone that has it. Some people with the allele show the trait, others don't, even though their DNA is the same at that locus. Environmental conditions are crucial in gene expression. For example, the gene that codes for the synthesis of melanin, the brownish pigment for hair, eye, and skin color, is only the instructions for producing melanin. You also need the building blocks of the protein that comprises melanin, like the amino acid phenylalanine. So without adequate nutrition, supplying your genes with the materials to build the proteins they code for, some traits may fail to develop normally. More broadly, no gene can produce its effect in isolation from the environment, and no environment can produce a trait without genetic instructions defining what cells can do with nutrients, hormones, and other signals. Think of it like building a house. The blueprints are your genetic information. The wood, steel, nails, windows, etc., they're the environmental inputs such as nutrients and hormones. You need both to end up with a structurally sound house, or in our case, a particular phenotype.
'Tis the season of gifting and halls to deck, and the Who's and Who Newville were in love with new tech. Where can we find Sonos and Samsung and Nintendo, they shouted. Would they find it in one place? This they questioned and doubted. When suddenly a Who yelled, Walmart's the place to start! And each Who added headphones, TVs, and games to their carts. With Walmart, their shopping was done in a flurry. They cried out, Who knew? And ordered their gifts in a hurry. Shop the latest tech gifts in the Walmart app.
Here for the Lowe's early Black Friday deals? You're right on time for some of our biggest savings. We're talking up to 50% off select major appliances, plus up to an extra 25% off when you bundle select major appliances. Holiday lights going up soon? Select ladders are up to 50% off right now. Get Black Friday prices without the Black Friday crowds. Lowe's, we help, you save. Valid through 11/19. Selection varies by location. Select locations only. While supplies last. See Lowe's.com for more details.
At Capella University, learning online doesn't mean learning alone. You'll get support from people who care about your success, like your enrollment specialist who gets to know you and the goals you'd like to achieve. You'll also get a designated academic coach who's with you throughout your entire program. Plus, career coaches are available to help you navigate your professional goals. A different future is closer than you think with Capella University. Learn more at capella.edu.
You may have heard the term epigenetics recently. This is changing gene activity without changing DNA sequence. Epigenetics has become a very, very big topic in genetics. For many years, it was common to think that the only way gene expression could change in a lasting way was through a mutation, a change in the base sequence of DNA. Now we know that there is another layer of regulation, and that's what we call epigenetics. Epigenetic mechanisms can activate or silence genes without altering the underlying DNA sequence. They do this through changes in how DNA is packaged and accessed in the cell. Key epigenetic processes include chromatin remodeling, which is altering the way DNA is wound around histone proteins, making some genes more or less accessible. Histone modification, adding or removing chemical groups from histones, such as acetyl, methyl, or phosphate groups. This influences how tightly DNA is wrapped and how active certain genes are. Non-coding RNAs. These are RNA molecules that do not code for proteins but can bind to DNA, RNA, or protein complexes to regulate gene expression. And DNA methylation. The addition of methyl groups to certain bases in DNA, especially cytosine. Methylation often has a gene silencing effect. Epigenetic changes are fundamental to normal development. All the body's cells, with a few exceptions, have nearly identical DNA. Yet liver cells, neurons, muscle cells, and skin cells look and function very differently. They do this by turning different sets of genes on or off, largely through epigenetic mechanisms. Sometimes, however, epigenetic changes contribute to disease. Aberrant DNA methylation or histone modifications can inappropriately silence genes that protect against cancer or activate genes that drive cell proliferation. Disorders such as obesity, diabetes mellitus, heart disease, and cancer have been linked to abnormal epigenetic patterns. One example of a condition with epigenetic involvement is Huntington's disease. Huntington's disease is a rare, inherited genetic disorder involving histone modifications that cause the progressive breakdown of nerve cells in the brain, leading to motor, cognitive, and emotional problems. It is inherited in an autosomal dominant fashion, meaning a child of an affected parent has a 50% chance of inheriting the gene. Symptoms typically appear between ages 30 and 50, and while there is no cure, treatments can help manage the symptoms. A particularly remarkable aspect of epigenetics is epigenetic inheritance. In some cases, changes in gene expression patterns can be passed from one generation to the next without any change in DNA base sequence. Environmental factors like diet, smoking, exposure to pollutants, chronic stress, infections, even famines can leave epigenetic marks on germ cells that influence how genes are expressed in offspring and possibly even grandchildren. In this expanded view, heredity includes not only the DNA sequence we inherit, but also the epigenetic modifications that shape when and how genes are used. Okay, I think that is a lot of genetics for A&P. For anatomy and physiology at the 100 level, your freshman A&P courses, your pre-nursing courses, etc., I think that's a pretty decent amount of genetics. Obviously, if you're taking a college genetics course, you're going to go way deeper than I have, or quite honestly, than I can. So maybe someday I'll have a geneticist on who can talk about this at a little bit more depth if that's something people are interested in. But for now, I think this is probably as much as you'll need. I hope that you enjoyed that. I hope it helps with your understanding of how the human body works on that genetic level. And I hope you listen to Smarty Pants Wonders, that podcast I was talking about in the beginning of the episode. I put a link to it in the show notes so that you can find it pretty easily, and I think it's a fun little break. The episodes are pretty short. It's a fun little break to listen to something about some cute animals. Or some scary bugs like lanternflies. Okay, so thank you so much for listening. I can't do this without you. I really appreciate all the comments that people leave, all of the ratings and reviews, the emails I get. I can't tell you how much it makes my day when I see that stuff. So thank you once again. I appreciate it. I hope this is helping you get your B or better in A&P, and I will talk to you next time. Anatomy and Physiology Bit by Bit is a production of Minus 55 Media and is hosted by the Believe Network. Please take the time to rate my show and leave a review. You can also check out the content I post on Instagram and YouTube as @StudentHelp4AP. That's the number 4 AP. Special thanks to my family, Bucks County Community College, and McGraw-Hill Education, where your instructor can find Anatomy and Physiology Digital Suite, my low-cost tutor video-based digital learning solution for anatomy and physiology currently being used at several colleges and universities. If your instructor isn't using Digital Suite, let them know about it. The link is in the show notes.
Tis the season of gifting and halls to deck, and the Who's and Who Newville were in love with new tech. Where can we find Sonos and Samsung and Nintendo, they shouted. Would they find it in one place? This they questioned and doubted. When suddenly a Who yelled, Walmart's the place to start! And each Who added headphones, TVs, and games to their carts. With Walmart, their shopping was done in a flurry. They cried out, Who knew? And ordered their gifts in a hurry. Shop the latest tech gifts in the Walmart app.
At Capella University, learning online doesn't mean learning alone. You'll get support from people who care about your success, like your enrollment specialist who gets to know you and the goals you'd like to achieve. You'll also get a designated academic coach who's with you throughout your entire program. Plus, career coaches are available to help you navigate your professional goals. A different future is closer than you think with Capella University. Learn more at capella.edu.